For most of medical history, the trillions of bacteria living in our intestines were treated as little more than passive passengers, tolerated at best, occasionally blamed for digestive discomfort, and otherwise largely ignored by mainstream cancer research. That picture has changed dramatically over roughly the past decade, as increasingly sophisticated research has revealed that this vast internal ecosystem, collectively called the gut microbiome, plays an active and, in some cases, surprisingly significant role in how the body responds to cancer, how effectively certain cancer treatments work, and even in the underlying processes that influence whether cancer develops in the first place. This has become one of the most rapidly expanding areas in all of cancer research, spanning laboratory studies, clinical trials, and large-scale human population research. And while it's important to be clear from the outset that no bacteria or probiotic represents a cure or a guaranteed cancer treatment, the evidence connecting specific gut bacteria to meaningful, measurable differences in cancer-related outcomes has become substantial enough that major cancer research centers now have entire divisions dedicated to studying it. Today, we're going to walk through what scientists have actually discovered about this connection, which specific bacteria have emerged as particularly significant in the research, and what the current evidence says about supporting a gut microbiome composition associated with better outcomes. To understand why gut bacteria would have any relationship to cancer at all, a disease that in most cases develops in tissues located nowhere near the intestines, it helps to understand a few of the mechanisms researchers have identified connecting these two seemingly separate systems. The gut microbiome has been found to play an extensive role in training and regulating the immune system, starting from infancy and continuing throughout life. And since the immune system is directly involved in identifying and eliminating abnormal cancerous cells throughout the body, a process called immune surveillance, differences in gut bacterial composition that affect immune function can plausibly influence cancer risk and progression through this pathway alone. Beyond immune regulation, certain gut bacteria produce specific metabolic byproducts as they break down dietary fiber and other compounds, some of which have been found in research to have direct effects on inflammation, on the integrity of the gut lining itself, and even on hormone metabolism, all of which connect to processes relevant to cancer development. And in perhaps the most clinically significant discovery of the past several years, researchers have found that gut bacterial composition appears to meaningfully influence how well certain modern cancer treatments actually work, a finding with genuinely direct and immediate implications for how patients might be supported during treatment, not just for long-term prevention. Let's start with what is arguably the most well-established and clinically significant discovery in this entire field: the relationship between gut bacteria and the effectiveness of a category of cancer treatment called immune checkpoint inhibitors, a relatively newer class of immunotherapy drugs that work by essentially releasing a brake on the immune system, allowing it to more effectively recognize and attack cancer cells. These drugs have transformed treatment for several cancer types, including advanced melanoma and certain lung cancers. But researchers noticed something puzzling early on. Some patients responded remarkably well to these treatments, while others, seemingly similar in terms of their cancer type and stage, showed little to no response at all. Multiple research teams investigating this puzzle found a striking pattern. Patients whose gut microbiomes contained higher levels of certain specific bacterial species showed significantly better response rates to checkpoint inhibitor immunotherapy compared to patients with different gut bacterial compositions. Several studies identified bacteria within a family called Ruminococcaceae, along with specific species including Akkermansia mucinifila and various Bifidobacterium species, as being particularly associated with favorable immunotherapy response across different research groups studying different patient populations. This finding was significant enough that researchers moved beyond simple observation into direct experimental testing, and in a series of studies that genuinely captured attention across the cancer research field, scientists performed fecal microbiota transplants, essentially transferring gut bacteria from patients who had responded well to immunotherapy into laboratory mice, and found that these mice showed improved response to the same immunotherapy treatment, compared to mice that received bacteria from non-responding patients, providing much stronger evidence of a direct causal relationship rather than simply a coincidental association. This research has progressed to human clinical trials as well, with early studies testing whether transplanting favorable gut bacteria into patients who initially failed to respond to immunotherapy could improve their subsequent response. And while this research remains in relatively early stages, some of these trials have shown genuinely promising results, converting a meaningful proportion of previously non-responding patients into treatment responders. Beyond immunotherapy specifically, research has identified several other significant connections between gut bacteria and cancer that are worth understanding. One area of substantial research interest involves colorectal cancer specifically, given the direct physical proximity between gut bacteria and colon tissue. Researchers have identified certain bacterial species, including some strains of Fusobacterium nucleatum, that appear with notably higher frequency in colorectal tumor tissue compared to healthy colon tissue, and laboratory research has found that this particular bacterium appears capable of directly promoting the growth and progression of colorectal cancer cells through several mechanisms, including production of specific toxins that can damage the DNA of colon cells, and interference with the immune system's ability to recognize and attack developing tumors in that location. This research has been significant enough that some researchers have proposed using the presence of this bacterium in tissue samples as a potential prognostic marker, and it has opened up entirely new research questions about whether interventions targeting this specific bacterium might eventually play a role in colorectal cancer prevention or treatment, an area of active ongoing investigation. On the more protective side of this research, certain other gut bacteria have been consistently associated in studies with lower cancer risk or better outcomes across multiple cancer types. Bacteria that produce compounds called short-chain fatty acids, particularly one called butyrate, produced when certain beneficial gut bacteria ferment dietary fiber, have received substantial research attention. Butyrate serves as the primary energy source for the cells lining the colon, and research has found it also has direct effects relevant to cancer prevention, including reducing inflammation within the colon, supporting the integrity of the gut lining, which helps prevent harmful bacterial products from entering circulation, and in laboratory studies, directly influencing the growth and survival of abnormal colon cells in ways that appear to favor healthy cellular behavior over cancerous transformation. Bacteria known for producing meaningful amounts of butyrate, including species within the Faecalibacterium and Roseburia genera, have shown associations in population research with lower colorectal cancer risk, reinforcing the broader picture that gut bacterial composition genuinely matters for this specific and common cancer type. Given everything the research has revealed about these relationships, the natural and genuinely important question becomes, what can actually be done to support a gut microbiome composition associated with these more favorable outcomes? And this is where the research, while still evolving, has produced several consistent and actionable findings. Dietary fiber intake stands out as perhaps the single most well-supported intervention in this entire area of research, and the reasoning connects directly to what we just discussed about short-chain fatty acid production. Fiber, specifically the type that isn't digested by human digestive enzymes and instead reaches the colon intact, serves as the primary fuel source for the beneficial fiber-fermenting bacteria responsible for producing protective compounds like butyrate. Multiple studies examining dietary fiber intake and gut microbiome composition have found that higher fiber diets are associated with greater abundance of these beneficial short-chain fatty acid-producing bacteria, and separately, population studies have consistently found associations between higher fiber intake and reduced colorectal cancer risk. Specifically, a connection researchers increasingly believe is substantially mediated through this exact gut bacterial pathway, rather than representing a separate, unrelated mechanism. Diverse fiber sources appear particularly important based on current research, since different beneficial bacterial species tend to specialize in fermenting different types of fiber, meaning a varied diet incorporating vegetables, fruits, legumes, and whole grains, rather than relying on a single concentrated fiber source, tends to support a more diverse and robust population of these beneficial bacteria compared to a narrower dietary pattern. Fermented foods represent another area that has attracted substantial research interest, given that foods like yogurt, kefir, sauerkraut, kimchi, and other traditionally fermented foods contain live bacterial cultures that can directly introduce beneficial species into the gut, alongside compounds produced during the fermentation process itself that appear to have independent beneficial effects. A notable study examining fermented food consumption found that a diet high in a variety of fermented foods produced measurable increases in gut microbiome diversity and reductions in certain inflammatory markers, findings that connect directly to the immune regulation and inflammation pathways discussed earlier as relevant to cancer-related outcomes. Though it's worth noting that research specifically connecting fermented food consumption to cancer outcomes directly, as opposed to these relevant intermediate markers, remains an area still actively being developed compared to the more extensive research base behind fiber intake specifically. Polyphenol-rich foods, the same broad category of plant compounds discussed in relation to direct antioxidant effects in other contexts, have also been found in more recent research to have a significant and somewhat underappreciated relationship with gut bacteria specifically. Research has found that many polyphenols aren't fully absorbed in the upper digestive tract and instead reach the colon, where gut bacteria metabolize them into smaller compounds, some of which appear to have their own beneficial biological effects, while this metabolic process itself appears to favor the growth of certain beneficial bacterial species over others. This research has led some scientists to describe polyphenol-rich foods, including berries, green tea, and colorful vegetables, as having effects that work partly through this gut bacterial pathway, in addition to whatever direct effects these compounds have elsewhere in the body, adding another layer to why diets rich in diverse plant foods show such consistent associations with favorable cancer-related outcomes across so many different areas of nutritional cancer research. On the other side of this equation, research has identified dietary and lifestyle factors associated with less favorable gut bacterial composition, and understanding these matters just as much as knowing what to actively include. Diets high in processed foods and low in fiber have been consistently associated in research with reduced gut microbiome diversity and lower abundance of beneficial, short-chain fatty acid-producing bacteria. Excessive alcohol consumption has similarly been linked in research to disruption of healthy gut bacterial balance, alongside its other, more directly established cancer risk connections, discussed extensively elsewhere in cancer prevention research. Unnecessary antibiotic use represents another significant factor the research has identified, since antibiotics, by design, kill bacteria broadly rather than selectively targeting only harmful species, and studies have found that antibiotic use can produce reductions in gut microbiome diversity that persist for months or in some cases longer, reinforcing why appropriate, judicious antibiotic use, reserved for situations where it's genuinely medically necessary, matters not just for the well-known concern about antibiotic resistance, but for gut microbiome health specifically, as it relates to this broader area of cancer research. Physical activity, somewhat surprisingly to people unfamiliar with this research, has also been found to independently influence gut bacterial composition, with multiple studies finding that regular exercise is associated with greater gut microbiome diversity and higher abundance of certain beneficial bacterial species, including some of the short-chain fatty acid producers discussed earlier, independent of dietary differences between more and less active individuals, suggesting physical activity itself, through mechanisms still being actively researched, exerts some direct influence on this internal ecosystem beyond simply what someone eats. It's important to close this discussion with appropriate scientific perspective, since this remains a genuinely rapidly evolving field where enthusiasm sometimes outpaces the current strength of evidence, particularly when it comes to commercial probiotic products, many of which make claims not yet supported by the kind of rigorous research described throughout this discussion. The strongest evidence in this field currently centers on dietary patterns, particularly fiber intake and diverse plant food consumption, rather than isolated probiotic supplements, and the immunotherapy research, while genuinely exciting and increasingly well established, remains an area where fecal microbiota transplantation and other direct microbiome interventions are still primarily being studied within clinical trial settings, rather than representing established, widely available standard treatment. What the totality of this research does support, with genuine confidence, is that the gut microbiome represents a real and significant factor in cancer-related outcomes, one that responds measurably to dietary and lifestyle choices largely within an individual's control, and that supporting this internal ecosystem through a fiber-rich, diverse, plant-forward diet, Thoughtful use of fermented foods, regular physical activity, and judicious antibiotic use represents a genuinely evidence-backed contribution to overall cancer risk reduction, and for those currently undergoing certain cancer treatments, potentially even to how well those treatments work. A finding that has fundamentally changed how researchers think about the relationship between what happens in our gut and what happens throughout the rest of our body. It's worth touching on how researchers actually measure and study something as complex as the gut microbiome, since understanding this helps explain both the excitement and the appropriate caution surrounding this field. Modern microbiome research relies heavily on a technique called genetic sequencing, which allows scientists to identify and quantify the thousands of different bacterial species present in a stool sample without needing to culture each one individually in a laboratory, a method that would be impractical given how many gut bacterial species cannot even be grown outside the human body under current laboratory techniques. This sequencing revolution over the past 15 years is largely what has made the depth of current microbiome research possible at all, but it also means that much of this research identifies associations between particular bacterial populations and particular outcomes without always being able to fully establish the underlying mechanism, which is why the mouse transplant studies and emerging human clinical trials described earlier carry such particular scientific weight, since they move beyond correlation toward genuinely testing cause and effect. Individual variability represents another important dimension of this research worth understanding. Since no two people's gut microbiomes look identical, shaped as they are by factors including early life exposures, genetics, geography, long-term dietary patterns, and even factors like birth method and whether someone was breastfed as an infant. This variability is part of why researchers studying interventions like fiber supplementation or fermented food consumption often find that individual responses vary considerably, with some people showing dramatic shifts in their bacterial composition following a dietary change, while others show more modest changes. An area of ongoing research sometimes referred to as personalized or precision nutrition, which aims to eventually predict how a given individual's unique microbiome will respond to specific dietary interventions, rather than relying purely on population-level averages, the way most current guidance still does. Taken together, what emerges from this rapidly growing body of research is a genuinely compelling picture, one where an internal ecosystem long dismissed as biologically peripheral has turned out to be an active participant in some of the most significant processes involved in cancer development, prevention, and treatment response. The practical guidance that follows from this research remains refreshingly consistent with broader dietary wisdom, prioritizing diverse, fiber-rich plant foods, incorporating fermented foods thoughtfully, staying physically active, and using antibiotics judiciously. But the depth of mechanistic understanding now available, down to specific bacterial species and the exact compounds they produce, represents a genuine scientific advance, one that continues to open new research questions and, increasingly, new possibilities for how gut bacteria might eventually be deliberately harnessed as part of cancer prevention and treatment strategies in the years ahead.